Hello everyone, and thanks for joining us today. Welcome to our latest Tecan webinar, Drug-Drug Combinations Made Simple. I'm Ashley, Managing Editor for Technology Networks, and along with my colleague Louise, we'll be moderating this event. We are joined today by three speakers. We have Dr. Roderick Beijersbergen and Dr. Pasi Halonen of the Netherlands Cancer Institute Robotics and Screening Center, as well as Jessica Merlino from Tecan. So screening drug combinations in vitro has become more and more interesting to researchers as they search for the best treatments for many diseases, which includes cancer. At the Netherlands Cancer Institute Robotics Screening Center, the NRSC, potential drug combinations that either restore or enhance clinical response are selected based on large-scale genomic screening projects combined with genomic data sets. The Institute has implemented an accurate, flexible, and user-friendly experimental setup for these combination studies by using the HP D300 Digital Dispenser. The NRSC uses an advanced technological platform to perform large-scale, high-throughput screening projects with cell-based or biochemical assays. These screens enable researchers to identify novel targets for cancer therapy and understand the mechanism of action of novel drugs, as well as to identify resistant mechanisms and determine effective drug combinations for specific patient groups carrying defined genetic alterations. In this presentation, the NRSC's workflow for identification of drug combinations and follow-up drug synergy studies using the HP D300 will be presented. Following the webinar, we'll have a Q&A session, and we would welcome any questions that you may have. To ask a question, you should click the Ask a Question button, which is underneath the media player window. Simply enter your email address and question and click OK. We'll aim to answer as many questions as possible during the time available. If you experience any technical issues, please use this button to request support. So our first speaker today is Jessica Merlino. Jessica is product manager at Tecan and has been there for 7 years in a variety of roles, previously working in biotech with her research focusing on cellular biology. Jessica will begin today's session with a bit of background on the HP D300 Digital Dispenser. So with that, I'll hand over to Jessica.
Hi, everyone. Thank you for joining our session today, where we will discuss drug-drug combinations made simple. As already mentioned, my name is Jessica Merlino, and I'm the product manager for the HP D300 at Tecan. Tecan now offers an instrument called the HP D300, which will make the setup of small molecule drug combinations very easy. To begin, I'd like to give some background information on the HP D300. First, I'll explain the HP-Tecan relationship, then the challenges with traditional titrations as described by many of our customers, and then the solution offered by the HP D300, finishing up with a conclusion. Hewlett-Packard and Tecan announced a partnership in March of 2011. HP manufactures the D300 Digital Dispenser and the cassettes, which utilize proven inkjet technology. On the other hand, Tecan sells, distributes, markets, and services the HP D300. Our customers have reported to us several common problems with their current titration method, including time-consuming methods, substantial waste both of compounds and consumables, risks, for example, with accumulated pipetting error, and inflexibility. We see that often these challenges lead labs to decide to skip certain experiments, such as drug-drug interaction. Here we show a traditional titration workflow. First, the user would set up their experiment. Next, there might have to be drug formatting. Maybe that's moving their compounds from tube to plate, for example. Next, diluent and plate preparation. Next step would be for the actual serial dilution to be prepared. The next step would show either having to tape or untape, mix, shake, spin, sometimes even pass off the serial dilution off to another group. There could be an intermediate dilution or in media or an aqueous solution. And finally, the possibility to prepare daughter replicates. In the end, then, that bioassay series would be used, that titration would be used in a bioassay series. On the other hand, using the HP D300, we would go right from setup, using the HP to actually dispense your compounds directly into your plate, and finally setting up the bioassay with that titration series. So what is the HP D300? The D300 is a complete solution. It includes the actual dispenser itself. It includes a powerful and easy-to-use software package, and also the T8 or D4 cassettes, which are used to actually do the dispensing. To use the instrument, a user creates a plate layout for the desired compounds in a plate and then follows the prompts to load the plate. First, the cassette is added into the instrument, and a few microliters of stock compound are pipetted into the cassette, as shown here. The cassette then dispenses directly into your bioassay plate using 22 nozzles. 2 of those nozzles dispense 13 picoliter droplets, and 20 of those nozzles dispense 20 picoliter droplets. A specific number of droplets are dispensed into a plate, building up to a final required dose. This eliminates the need for serial dilution. Some further details we can see here. For instance, here you can see an ejection of a 20 picoliter droplet directly from one of those nozzles in the dispense head. And then down below, we can see that depending on the dose requested in each well, here we can see a 13 picoliter droplet dispensed. And here for a 40 picoliter dose, we see 2 20 picoliter droplets. Those number of droplets would build up to whatever the final dose is, even into the microliter range. So multiples of 13 or 20 picoliter droplets are dispensed into each well. And this is done really quickly because thousands of droplets are dispensed per second. Any dose can be dispensed into any well, and you can see here the compatibility for the well plates. 12 well up to a 1536 well plate can be used, and those can be either assay ready, meaning already containing all of the reagents needed for an assay, or dry. There's also an optional plate shaking. This is optimized for use with small molecules and DMSO. Here you can see an overview of the software that can be used. It really is easy to learn and easy to use. You can see here that there are colorful graphics to specify the plate layout, making it easy to see at a glance if it's been set up as desired. Also, the software will calculate the required dose needed within each well, depending on the concentration requested within that well. There's a variety of wizards that are available to make setup even faster. And the software will direct the user through the process, when to pipette, when to load cassettes and plates, etc. I'd like to go through, finally, to show some of the different experiments that can be enabled by the D300. Of course, the focus of this talk is on drug-drug interactions, which we can see here, and that's can be set up using even a wizard called the Synergy wizard. Next, I'll talk briefly about some of the other options, though. The next one would be targeted titrations, randomization, and even high-density titrations. The drug-drug interaction experiment is especially easy to create with the HP D300 because each dose of compound is dispensed in a contactless fashion and directly into each well, independent of any other doses or wells. Setup for this experiment with traditional serial dilution is possible, of course, but it can be tedious and very time-consuming. The HP D300 software even has a wizard available for experimental setups of 2 compounds or even more titrated against each other. Here down at the bottom, we see one example showing compound A and B, which are titrated across the plate. So there's compound A and then compound B. And then compound C in the same plate is titrated down the plate, resulting in a nice interaction experiment of compound A versus C and compound B versus C. Our speakers from NKI will discuss the use of this feature further in their talk. A second type of titration that can also be set up with a wizard is called a targeted titration. Here we can see an example of a typical titration where each dose is laid out evenly spaced across the entire dosing range. However, in some cases, it could be interesting to cluster doses around a specific value, which leads to a more precise determination of IC50 values, for instance. So here we have a couple of doses at each end of the curve. We have a couple of doses at the high and the low end of the curve. And in the center, we have the doses that are clustered. Next is randomization. Especially in cell-based assays, plate edge effects can cause problems. So to avoid this, some users have found that they just stop using the very outside wells of the plate, the edge wells, something like that. That can waste up to 39% of a 96-well plate. By using the randomizing function, some users have found that they can again use the entire plate since the systematic bias of sample location is reduced. So here, to take this first sample layout and turn it into the second plate layout, we can actually just use the click of a button to randomize those. There's a report file generated with each plate run, which shows both the randomized and non-randomized location of each well, and this can help with data analysis. Finally, an extreme example of the use of the HP D300 shows here a high-density titration. This is actually one compound that was titrated into 384 wells, creating 384 doses of that compound. It results in more than 50 data points per log and a very high precision data in the sloped region of the curve, which we can see here. Of course, this would be completely infeasible to set up with traditional methods, and probably a really extreme example that not every user would set up on a daily basis, but it shows the power of the D300 nonetheless. So, in summary, some of the D300 benefits include accurate drug-drug titration made possible, randomization, as we had talked about, targeted dosing, giving you really precise IC50 values, high-resolution titrations, giving you more than 50 data points per log, and also an extended working range between 10 microliters down to 13 picoliters. This direct titration eliminates the need for serial dilution, and it also gives time savings within a lab. Because any dose can be added into any well of an assay plate, it makes it super flexible. And finally, there's virtually no cross-contamination risk between wells.
Thank you, Jessica. For our next speakers, we will now move on to the NRSCs, Dr. Roderick Beijersbergen and Dr. Pasi Halonen. Dr. Beijersbergen is an associate professor in the Division of Molecular Carcinogenesis and the head of the NKI Robotics and Screening Center. He obtained his PhD in cell cycle regulation in 1996 at the Netherlands Cancer Institute. During the last years, his work involves the design, construction, and application of large-scale RNAi interference libraries to perform loss-of-function genetic screens in mammalian cells. He's developed the novel screening method called shRNAi screening. This technology is applied to identify the mechanism of action of anti-cancer drugs and genes involved in resistance. He is also head of the NKI Screening and Robotics Center, where large-scale automated cell-based assays using high-content imaging for complex phenotypes are performed. With these technologies, he aims to identify synthetic lethal interactions in human cells with the goal to identify novel drug targets for the treatment of cancer and other disease. Dr. Halonen is a postdoc division of molecular carcinogenesis at the NKI Robotics and Screening Center. He obtained his PhD in enzyme kinetics and protein structure-function relationships, studying inorganic pyrophosphatase, a ubiquitously expressed enzyme in energy metabolism at the University of Pennsylvania in Philadelphia and the University of Turku, Finland in 2005. During the last years, he worked at the Robotics and Screening Centre in the Netherlands Cancer Institute to develop, execute and analyse the siRNA and drug assays with spectroscopic as well as protein and image-based readouts in collaboration with the researchers at NKI. Most recently, he's been developing novel tools for drug synergy studies, as well as 3D mammalian cell culture models from patient-derived samples to rapidly identify and test more efficient drug combinations of critical clinical relevance. So with that, I will hand over to Roderick to begin his presentation. Thank you.
Okay, so today we'd like to discuss our experience with the work on using the HP D300 Digital Dispenser in our work for drug-drug combination screens. So the focus of our research is really in using functional genomic technologies to identify and predict effective combination cancer therapies. And today we'll highlight a few examples of how we're trying to identify effective treatment combinations, and at the same time illustrate this with the use of the digital dispenser in our high throughput work. Nowadays there's a general consensus that we need to use genomic data to sort of stratify patients or to identify groups of patients that are responsive to a specific kind of cancer therapeutic. Of course, also these days there's a strong focus on the application of targeted agents in cancer patients that make use of specific genetic alterations that are present in these patients that then would be eligible to such a specific or personalized treatment. However, a major challenge is to identify these genetic alterations in such a way that they can be broadly exploited in cancer patients. And of course, on this slide is illustrated already that in the case of squamous cell lung cancers, the type of mutations are quite diverse. The frequency of strong driver mutations can be really low, even within this specific subgroup of tumor types. And therefore, it makes it difficult to identify those targets that can be effectively explored for therapy. However, it also became clear that when we find these different mutations present within a single tumor type, it's also clear that they can cluster along pathways. And these pathways, of course, are involved in the regulation of important biological processes. And the understanding of these pathways can make us sort of move forward in identifying the weak spots within these pathways that can be, again, exploited in cancer patients. However, in many cases, it's difficult to predict whether a patient will respond to a given therapy that's targeting one of these specific nodes. And actually this is nicely illustrated by an example that we have exploited in colorectal cancer, where a small group of patients actually carries a mutation in the BRAF oncogene. So the classical mutation of BRAF, which is a kinase that's involved in the MET kinase signaling pathway, is found in melanoma. So almost up to 80% of melanoma patients carry this specific BRAF V600E mutation that makes it a constitutive active kinase. And therefore, when this pathway is constitutively active, it drives proliferation. Well, now it's striking that the same mutation of BRAF V600E is also found in colorectal cancer, although with a much lower frequency. And interestingly enough, in the last few years, a very effective drug called Plex, of nowadays vemurafenib, has been developed to inhibit this kinase. However, when this kinase inhibitor is applied in melanoma patients, we see a very strong response, as depicted here, but more than 80%. But when this is applied in the same group of colorectal cancer patients that do carry the mutation, we see a very low response rate, although we find the same mutation and the same activation of the pathway. So this striking difference really led us to look into this further and trying to identify a potential mechanistic explanation for this ineffectiveness of this therapy and potentially a future combination therapy that would be far more effective in these patients. To identify these interactions, we actually make use of functional genomic technologies that we've been using for the last 10 years in our lab to identify genes that then can enhance the response to this inhibitor. So the first challenge, of course, is to replicate what we see in the patients in cell line models. What you see here in this slide is actually cell line models based on colorectal cancer cell lines or melanoma cell lines that are treated with this vemurafenib or Plex inhibitor of BRAF and then look for their survival in a short-term assay. So what you see in the blue line, this is a melanoma cell line, is highly sensitive to this drug, whereas a panel of colorectal cancer cell line carrying the BRAF mutations are far less sensitive to this drug. And this can also be seen in a long-term growth assay where you see a strong reduction in cell proliferation in the melanoma cells, but there's hardly reduction in the colorectal cancer cell lines. So really the vemurafenib or Plex drug is ineffective in BRAF mutant colorectal cancer, where it's highly effective in BRAF mutant melanoma. Well, to identify a potential players in this pathway, we use our so-called barcode screening system, where we can screen very large collections of vector-based shRNAs in a large population of cells, and then to identify those hairpins that actually enhance the response to the treatment with vemurafenib. So to do so, we take again a colorectal cancer cell line carrying the BRAF mutation, the wither cells in this case. We infect with a large collection of shRNAs split in two identical fractions, of which one is treated with the vemurafenib inhibitor. Then we allow them to grow, and now there will be selection for those hairpins that enhance the response to vemurafenib in this branch, and they will be depleted from the population when we look around 10 days later. These hairpins will not be depleted from the control because they don't have an effect by themselves, and therefore we can identify those hairpins that enhance the response. So a typical result of such an experiment is seen in the graph on the right, where we now look at all hairpins and we look for their relative level of enrichment when we compare the Plex vemurafenib treated population to control. And now we see that there's a small number of shRNAs that what we call drops out of the population, indicating that those hairpins are negatively selected in the population treated with vemurafenib. And interestingly, in this population we find 3 independent hairpins targeting EGFR, indicating that the inactivation of EGFR receptor expression in cells treated with vemurafenib enhances the response in these colorectal cancer cell lines. So to continue on this a little bit further, we demonstrated that indeed knockdown of EGFR receptor, indeed on the left, shows that the cells stop growing. We have very effective knockdown on the protein level. And now interestingly, of course, in the clinic, we've already developed a large class of inhibitors of EGFR signaling, among which are cetuximab and gefitinib. So then we actually tested whether the combination of BRAF inhibition together with EGFR inhibition would be more effective than either treatment alone. As I indicated before, inhibition of BRAF alone in these cancer cell lines hardly has an effect. And also only treating with cetuximab in the absence of Plex, there's no effect, or if gefitinib, there's no effect. But we see a striking synergy in the combination of these two drugs. So really we can restore sensitivity of BRAF mutant colorectal cancer cell lines by combining a BRAF inhibitor together with an EGFR inhibitor. And of course, for this presentation, it's not essential to go into the details, but really what we figured out is that feedback signaling is really essential in these cancer cell lines, how they control either excessive signaling or disturbance of signaling pathways. So what happens actually in cells that have a mutation in BRAF, when you block this mutation, normally when this is active, it will cause activation of downstream targets, and then there's a negative feedback that then inhibits the upstream pathway. However, when you have a RAF mutation, then this pathway is highly active and there's a lot of feedback signaling. But now if you add a drug against the RAF mutation, you block this pathway. As a result of this, the activity of the downstream factors go down, you lose feedback activation, and as a consequence there will be reactivation of these growth factor receptors and then reactivation of the downstream pathway that can then either reactivate the signaling pathway or it can start to activate other parallel pathways, among which is the PI3 kinase pathway, that actually now then again allow bypass of the inhibitor of RAF alone. So really it's upstream activation after inhibition downstream that causes the resistance to this treatment. And we actually believe that this type of regulation will be crucial for the implementation of these targeted agents within signaling pathways, because we have to understand these feedback loops and potentially perturb these feedback loops to have an effective therapy in the clinic. And that this does not only work in cell lines, we also showed actually in xenograft models, where here we take xenografts of these BRAF mutant colorectal cancer cell lines, and we show that only the combination of EGFR inhibition plus BRAF inhibition is effective in these models, where single agent treatment, either BRAF with this, vemurafenib or only EGFR inhibition with cetuximab, is without effect in these models. Of course, this immediately from a screen highlights the potential to use this in the clinic in those colorectal patients that carry a BRAF mutation. And indeed, this is a clinical trial that's ongoing at our institution and until now seems to be quite successful. Of course, it was quite unexpected to identify one of these upstream mediators in the pathway's potential resistance mechanisms. And so what we now know, of course, is that there are many, many cancer drugs out there, and then of course estimations go to up to more than 1,000, that can be potentially be combined as more effective treatment therapies. And of course now we then think that we can use functional genomics to drive sort of a pre-selection of these compounds that can work in synergy, and then go into detailed studies to try to elucidate their mechanism and potentially try to identify let's say, a combination that can be applied in the clinic with relative ease, because these single drug agents are mostly already tested through phase 1 or sometimes even phase 2 trials, and so therefore they don't need a long lag phase before they can actually implement it in the clinic. However, of course, we do appreciate the idea that we're looking at thousands of possible combinations that need to be explored to identify those that are most effective in the clinic. And for this, of course, we use an integration of functional genomic technology with high throughput platforms to allow us to do this kind of work. And Pasi actually will now start to discuss a little bit how we use the HP Digital Dispenser in our work in a sort of high throughput fashion to identify the drug-drug interaction mechanisms.
Okay, so this is basically what half of our high throughput screening laboratory looks like. We have integrated and automated pipetting stations, robots there working in different types of screens, and we have integrated incubators and plate handlers into our workflows, and here we do all of our compound screens and then functional genomics screens. Now, previously we used to make all the compound dilutions, for example, for IC50 determinations using the robots, but our workflow was greatly reduced and aided by the HP D300 Digital Dispenser. And here are some of the properties that Jessica just described, and it definitely has speeded up our workflow and assays we're conducting here. When we first started using the HP D300 drug dispenser, we first wanted to check certain really basic things if they work. For example, if the digital dispenser works in a similar fashion to our robotic platform, and also some basic ideas we were thinking that might affect assay quality and readout quality. So, for example, in this figure, we have a 384-well plate where we have added drugs in two different ways. For example, here in this top part of the graph, we have an ordered addition of drugs, and we have the same set of drugs here in the lower part, but here the drugs have been randomized. We do a very basic proliferation assay, and after 3 days we measure the proliferation or cell viability using cell-titer blue reagent. I have plotted the data here in this lower graph, and as you can see, it doesn't really matter if the drugs have been added in an ordered fashion or a randomized fashion. The data is similar in both cases. We also tested if drugs can be added prior to cells or after the cells. The data looks very similar in both cases, and there's a slight difference in the determined IC50s depending if the drugs have been, depending on the order of drug addition. But all in all, the results are quite reproducible, and the fitting can be performed quite easily later on in an external software. Now, if we start using more and more drugs, we can, of course, determine the IC50s in a matrix type of fashion. So we titrate drugs in two different directions, and then we can calculate the IC50s of one drug in the presence of the other drug. These are the two different drugs that have been used in this example. But of course, this produces quite a lot of different IC50s, and there's nothing wrong with that. But we thought that maybe there's a different way of analyzing the data, and that's why we came up with a new kind of solution using a so-called synergy tool for the NKI Research, Robotics and Screening Center. And what we do in this approach is that we have a 384 or 96 well plate. We have different matrices on the plate where we titrate drugs in two different directions. And for example, in this plate layout, we have 6 different matrices, and we have several different drugs in this matrix. And we also have positive and negative controls for our assay. Plate can be incubated for a short time or a longer time, and we use the data from this matrix to analyze the results in a global fashion. And what this means is shown here. So we have one of these matrices shown here, and this is just experimental data. For example, degree of cell death. And we have 2 drugs in 2 axes. We have drugs where, when in these vertical and horizontal lines, as single doses or as single agents, and then in the remaining part of the matrix, we have them in combination. Now, using these single agent lines and columns, we can actually fit the data in a linearized fashion or in a sigmoidal fashion to get the IC50s. And using these single agents, we can model the data in a different kind of matrix, and we get a so-called reference model. Reference model in this case describes a system or a readout, what you would expect if there is no synergy between 2 drugs. And in pharmaceutical terms, this means that the combination index would be 1. Now, when we have the experimental data and we have the reference model, we can subtract the reference model from our experimental data, and we end up having a so-called excess matrix, which is just one matrix subtracted from the other. And now when we look at this final output, we can actually see certain pockets of synergy and certain combinations of drugs that produce these synergistic values, where the experimental effect has been higher than expected, calculated based on the model that we used as a basis of our analysis. So using these matrices, we can do experiments in a short term, as shown here. So we have an experimental data set here, and the excess matrix here, and we can compare the results to long-term experiments, as shown here in a long-term assay. Now, in this case, when we checked the activities of 2 different drugs, in short-term and long-term, the results were quite similar. So, for example, here, if we look at our synergy matrix, we see that the region where these 2 compounds produce the highest degree of synergy in short-term can also be reproduced in long-term, because here we're losing cells almost completely. When we look at different areas of the synergy matrix, we see less combined effect, and then they are also reproduced in our long-term assays, as shown by the green squares and the orange squares. And of course, in areas where there's hardly any synergy, the cells grow just fine, and they are not affected by the drugs in combination. So, in this way, using the synergy approach and the matrix approach, we were able to verify the results also in long-term assays, and we decided that we can use them also later on in other types of assays and in a larger scale.
So having established a nice correlation between, indeed, short-term assays as generated by the HP Digital Dispenser compared to our long-term assays, of course, has motivated us to do again an example of a potential drug-drug combination and see how we can explore this for cancer therapy. And along the same lines as the previous example that we discussed in colorectal cancer, there's also still a strong clinical need to identify inhibitors in those mutants that actually carry a RAS mutation. And this mutation, of course, is frequently found in lung cancer, but also in colorectal cancer cell lines. And as you probably memorized from the previous slides, we have the same pathway, the MAP kinase pathway, where now rather than having a RAF mutation, we have an upstream RAS mutation that drives the activity of this pathway. And of course, we have also small molecule inhibitors downstream of RAS, for example, MEK inhibitors. And one of these inhibitors is called sulometinib, which can be used to treat patients that carry a RAS mutation. However, similar to the treatment of colorectal cancer with BRAF inhibitor, we also see that many of these RAS mutant lung cancers do not respond to MEK inhibition, nor do the colorectal cancer cell lines. Although here we have one example that's highly sensitive to this inhibitor. So again, we asked ourselves, can we find potential drug-drug combinations that can be used or explored in the clinic? So without going through all the details, we again performed one of these pooled shRNA screens to identify those hairpins that drop out under treatment. We use here colorectal cancer cell line SW480, and we identify actually a gene that's represented by two individual hairpins that's targeting ERB3. ERB3 is one of the receptors out of the HER2 family, or the HER family, that consists of EGFR, ERB2, 3, and 4. Indeed, when we knock down this ERB3, we see a strong enhancement of the response towards the MEK inhibitor sulometinib. So, of course, now it would be relatively simple to say, well, we inhibit ERB3 together with the MEK inhibition, and then we get a strong synergistic response. However, as I said before, these are part of a whole gene family, so the question is which of these receptors should we target to get the strongest synergy? A inhibitor that inhibits actually all receptors, one that's specific for EGFR, or actually a combination of EGFR and ERB2, or specific ERB2 inhibitors. So of course here we have a need for a high flexible platform that allows us to test many different combinations of compounds in a relatively short period of time to really drive the decisions how to explore this further. And of course, really the HP Digital Dispenser is very useful for this. So again, as Pasi already discussed, we create these dose response matrices of two different combinations. And in this case, of course, we use drugs that either target MEK, which is trametinib or silumetinib, also known as HD6244, in combination with inhibitors that target components of the HER family. And then, of course, we do the same short-term experiment where we plate in 384 well plates format. We have drug addition by the digital dispenser. Then we add the tyrosine kinase inhibitors from the HER family. And then we wait for 96 hours and read cell proliferation in this assay to identify the most effective combination. And what we show here is an example where we use these two drugs. So this is afatinib, which is a pan-ERB inhibitor. So it inhibits all members of the family together with the MEK inhibitor, and here you see that either trametinib or another drug AZD, there you see that the combination is highly synergistic and we get this synergy score. This is really high, 3.27 and 3.24 in these two combinations. So again, very rapidly making the transition of a functional genomic screen, having the access to a very flexible platform that allows you to rapidly identify drug-drug combination matrices and from that identify the most potent combination. And because we now know, of course, that ERB3 apparently plays an important role, we also can explore this as a potential predictive marker to the response of the combination. Because what we see that in cells that do not express or at a very low level, they actually do not respond to the combination, whereas cells that do express ERB3 are responsive to the combination. And this is again illustrated here by one of these synergy experiments performed with the HP Digital Dispenser, whereas the H1792 cell line that's depicted here has hardly any ERB3 expression, doesn't show synergy, score of 0.58, whereas in the SW837 that has high expression of this ERB3 receptor, depicted here, we see very strong synergy. And over a large panel of cell lines, we see a very good correlation and significance difference between those cell lines that have no ERB3 expression compared to those that have high ERB3 expression with respect to the synergy that we can achieve with this combination. So again, with this, of course, we can now pursue further and show also in long-term assays now in KRAS mutant cells. So here it's in H358 cells that really see that the inhibitor MEK plus this ERB3 inhibitor or the pan-ERB inhibitor, we see very strong synergistic responses, which can be now also explored in the clinic, a potential drug combination in RAS mutant, either colorectal or lung cancer cells. And of course, we show here this in H358 KRAS mutant cells, but also other cell lines that carry KRAS mutations are highly sensitive to the combination that we identified through these drug-drug interaction studies. And again, so when you look in these cell lines, you see this discussed before, this very nicely combination and synergy. But in other colorectal cancer cell lines, HT116, that also carries a KRAS mutation, we don't observe this synergy. But we also know that in this cell line, actually, they also carry a PI3 kinase mutation. So presumably, the activity of the PI3 kinase can actually counteract the combined inhibition of the MEK ERB3 in these cell lines. And therefore, of course, you need to direct towards another combination. So again, illustrating that there's a need for a high flexibility of a platform where you can rapidly screen drug-drug combinations through many different cell lines in different concentrations and different combinations. And the HP Digital Dispenser, of course, is an excellent tool to perform these studies. So at this moment, this is what we're doing. We're generating many of these dose response matrices with different kind of compounds, either targeting PI3 kinase pathways, mTOR, MEP kinase, ERK pathway, or upstream RTKs to identify their potential synergy in different cell line models carrying different genetic alterations, and so therefore being able to either stratify patients based on their genomic alterations, and in addition identify dose-effective drug-drug combinations that can then be used specifically in those patients that carry specific genetic alterations. So I think in summary we can conclude that really the HP D300 digital dispensing systems really accelerates the follow-up and validation of these drug-drug combinations that we identify in our large-scale functional genomic screening projects, that this is a highly flexible and accurate platform that we can have easily adapt to different compounds and combinations, but also different concentrations, and importantly, also be used in different cell types using a variety of readouts. And therefore, this really generates this very efficient follow-up sort of strategy for these functional genomic screening projects. And apart from that, of course, the D300 also saves valuable reagents and costly and time-consuming dilution ranges that would normally do on our large-scale robotic platforms, and thereby really accelerates the experimental design and setup and limits the hands-on time for individual researchers. And also I think that the platform is relatively easy to use so that individual researchers can actually program and design their own experiments without being a need for supervision from skilled either technicians or operators. Okay, so with that I'd like to end, and I'd like to acknowledge the following people that contributed to this work. Really at the NKI Screening Center, it's Pasi, Halonen together with Cor Liefding, who developed the complete drug synergy matrix model, together with Ben Morris, the technician who's really driving the programming and control of the robotic systems. Many of these projects are in close collaboration with the lab of René Bernard, where really Chung Sun did most of the experiments that we discussed today. And also the other members of the labs and our microarray and sequencing facility, the bioinformatics groups, and our international collaborators for the validation and follow-up experiments are greatly acknowledged on this slide. And with that, we'd like to end, and thank you.
Well, thanks to Jessica for that most interesting presentation. My name's Louise Conlin, and I'll be hosting this Q&A session. On the line with me today, I have Jessica Merlino, who is the product manager D300 at Tecan. I also have Roderick Beijersbergen, group leader, and Pasi Halonen, who is the postdoc researcher, both from the Netherlands Cancer Research Institute in the Robotics and Screening Centre. Right, so our first question that we've had in is how many people in your lab are using the instrument, and that's to the Netherlands Cancer Institute, please.
So we have approximately 75% of our personnel on this floor, which makes 15 to 20 people, but because it's an institute-wide use for the instrument, I would say maybe like 30, 35 people on a regular basis are using the machine.
That's great, thank you. Next one we have in is, do you have any concerns about dispensing high concentrations of the MSO directly to cells?
Until now, we have not experienced any of these problems. We believe that this is not really an issue because we dispense very small volumes of DMSO onto the medium of the cell so that it gets quickly dispersed into the solution. Of course, there's also the option of shaking in the machine itself, or we can shake the plates afterwards after all the dispensing. So until now, we have not seen any adverse effects of directly dispensing DMSO to the cells.
If I could ask a quick question, Roderick, are you using the plate shaking function in order to disperse the DMSO even quicker, or do you not shake the plates?
We sometimes use it, especially when we dispense a larger volume, so we can disperse it and dispense and shake it right away. But then we also have an external shaker. Usually then we do them in batches. But the option is there in the digital dispenser as well.
Okay. Yeah. That's great, thank you. The next one we have in is the compound has a low solubility. Is there a chance that the cassette can get clogged?
Okay, so in our experience, we, I think, routinely use concentrations of compounds that are less than 10 millimolars, and most of those compounds are highly soluble in DMSO. I have to say that we have seen problems with compounds that were not very soluble in DMSO, so there was some precipitate present within the solution, and then when that was used in the digital dispenser, we did notice some problems with the dispensing that could be related to clogging. But in general, we don't have this problem if the compounds are actually good dissolvable in DMSO.
Great, thank you. So another related question here, which has been touched on. Are there any influences of viscosity on compound solution for the accuracy of dispensing?
Well, so far we've only been using the DMSO solutions. It's standardized for the DMSO, so we haven't had problems with those. Of course, we're also interested in maybe using other type of solvents, but then it's up to the R&D at Tecan to maybe answer these issues.
Yeah, that's right. The instrument as it stands now is only for dispensing small molecules in DMSO. That's what the liquid handling properties of the cassettes are made for. Of course, I'm sure you can imagine lots of people ask about dispensing other fluids or items out of the dispense heads, but that is currently not an option.
Great, thank you. Another DMSO question here. Different DMSO per well for titrations. So does the D300 add DMSO to keep the DMSO constant?
So there's actually the option within the software to program backfilling or normalization of DMSO, and that can be done using either the standard T8 cassette, or you can also use the D4 cassette, as it's so called, for DMSO normalization, and that allows you to fill up to 200 microliters of DMSO in order to normalize. So it's done quickly using that relatively new cassette.
That's great, thank you very much. Next question we have is, what's the volume of each well holding one compound for dispensing, and how long does it take to dispense one 384 well plate layout from one dispensing cartridge?
So the standard T8 cassette can hold up to around 10 microliters of compound, and the D4 cassette, like I just mentioned, can hold just over 200 microliters of DMSO. And the time it takes to dispense into a 3D4 well plate from a single dispense cassette would be around 100 seconds.
That's great, thank you, Jessica. Next question we have, how many drug combinations can you analyze per week using this software?
So the software that we're using here is we've written a script for the analysis. So it pretty much depends on first of all, of course, the quality of the data. We can run quite a lot of the 384 well plates that we're using as the basis of our assay, and we probably could run more of them than we're interested in analyzing, because the amount of data that we can actually produce is quite. It's not now limited by the dispensing of the reagents. It's more like in the analysis. So we haven't really running these assays day and night because then we don't have enough people to analyze the data. But it definitely has allowed us to study, classes of drugs versus other classes of drugs in a very versatile fashion, and because then we can run the replicates and technical replicates, biological replicates in such quantities that when we actually do the analysis, we use an R script and then to analyze the data. It's the analysis itself is pretty fast also.
That's great, thank you. In here about the cleaning of the apparatus. What is the cleaning process for the nozzles on the D300, and is there a status option to assure that all nozzles are working properly?
So there is not a process for cleaning the cassettes, actually, because they are single use. The instrument actually tracks to know which dispense heads have been used, so that there's no risk for cross-contaminating by adding a second compound into a dispense head that's already been used previously. And there is a kind of check that's done by the instrument itself before the instrument is asking you to load compounds into each dispense head to ensure that that dispense head will be working properly. But they are single use.
That's great, thank you. We have a question in here, I think this is the last one for today, about undesirable side effects of drugs. This person was just wondering if you're planning any PK or PD or ADME studies to identify the feasibility of using such drug combos in patients.
Yeah, I think this is definitely a challenge in trying to identify these new combinations. Of course, until now we have been, I think, fortunate in the identification of combinations of drugs that were previously used in the clinic. So for these drugs, it was already known at what concentration there would be adverse side effects. To our surprise, actually, what we see at this moment is that when we combine two drugs, that we see more a reduction of the adverse side effects and the unexpected toxicity than that we see an increase in the side effects. I think one explanation for that, of course, is that because we use drugs in the same pathways, and many times also due to these feedback activation mechanisms, a single drug can cause aberrant activation of other pathways, resulting in these unexpected toxicities, whereas when you combine these two, you see a reduction. And this has already been described, for example, for the combination of a BRAF inhibitor and the downstream MEK inhibitor. So I agree, it is a challenge, but until now we're fortunate that we do not see this as a serious obstacle for combining these drugs in patients.
Right, thank you very much. That's all we have for today. Thank you for answering those questions so clearly. Right, to all attendees, if for any reason you or a colleague missed any or all of today's webinar, we're very pleased to say the on-demand link will be available on the Technology Network's ADME Talks community within the next 48 hours. There will also be an email sent out to everyone who registered, so be sure to keep an eye out for that one. So all that's left is for me to thank Jessica and all those at Tecan for making today's webinar possible. A big thank you to everyone who attended this presentation. We do hope that you found it enjoyable as well as informative. Thank you very much.